We've talked about the T cells and the B cells and the humoral, the cell-mediated divisions of the specific or adaptive immune system. Let's focus in on antibodies now. Antibodies are these Y-shaped structures. Antibody, remember, same as immunoglobulin, same as gamma globulin. They're glycoproteins. In other words, they're proteins that have sugars on them, shaped like a Y or a T. They have two heavy chains, two light chains. You can see there in the illustration, it shows the light chains and the heavy chains. They have constant regions and variable regions. So the constant regions are the ones that are the same for every antibody. Variable are going to be the receptors. That's where you can see in the illustration there, the variable, the V regions, that's where antibodies are going to be different from one another. So look in the lower right, you can see how an antibody, you see the antigens listed at the top there, the purple, the red, the green, the yellow, the blue, and the whatever, the Dijon, maybe, I don't know. But you can see that different antigens have differently shaped epitopes, and therefore the antibodies have differently shaped receptors. That's why every antibody only can respond to one antigen. See, if it doesn't have the right shape, it can't respond to it. See, so we see the antibody there. It has a receptor that's perfect for the yellow antigen. It would not match up with the purple antigen or the blue one or any of those other ones, and therefore that's why it can't do anything to those. It can only respond to the one that it's been designed for. They function as receptors on B cells, and they also freely float. So the receptors on B cells are the same. With the B cells that become plasma cells are going to take their receptor, and then that's what's going to become the antibody. An antibody can only bind the specific antigen it has been designed to recognize, which I was just saying. All right. So the variable regions, unique for each B cell or plasma cell that manufactures them, they can bind antigens causing neutralization, immobilization, agglutination, and precipitation. We talked about that in the last PowerPoint. It can cause all those things to happen. Stem of the Y cell is recognized by other immune cells and molecules. So natural killer cells, complement proteins, eosinophils, what they do is they wander through the fluids of your body. And when they see the constant region, all right, the C region, the heavy chain, you can see there in the illustration, when they recognize that, then they go after whatever is attached to the two variable regions. They'll kill whatever is attached to the two arms of the Y. Really cool. Allows foreign cells and molecules to be targeted for destruction. Antibodies, again, don't kill. They just handcuff the bad guys. And then the cells of your body, eosinophils, complement proteins, natural killer cells, recognize the stem and kill whatever is attached to the arms. Now, there are lots of different kinds of antibodies. The immune system makes billions of them. So the way that it does that is called somatic recombination. This was kind of a puzzle for a long time. You only have about 35,000 genes. And the basic rule of molecular biology is one gene, one protein. All right, it's like the motto of the Texas Rangers: one riot, one ranger. You got a riot, all you need is one Texas Ranger to take care of it. All right, but so if you only have 35,000 genes, how can you have a billion different, or more, perhaps billions, of antibodies? That was kind of a puzzle for a while. How can you make billions if you only have 35,000 genes? Because again, antibodies are proteins, right? Well, normally it's one gene, one protein. How can you make a billion different proteins if you got only 35,000 genes? Well, DNA segments are shuffled to form new combinations of base sequences to produce antibody genes. It's kind of like, you know, I said before, imagine a deck of cards. There are 52 cards in a deck of cards, all right? Now, does that mean then that there are only 52 sequences that you can lay out of cards? No. When you shuffle a deck of cards, you get, you know, a weird, you know, peculiar, individual arrangement. You know, you get the 2 of hearts, and then the 9 of spades, and then the jack of diamonds, and then the 4 of diamonds, and then the 6 of spades. And so what happens is when you shuffle the cards, even though there's only 52 cards, you generate many more than 52 sequences. So that's what happens in somatic recombination. Your body takes the 35,000 genes and basically shuffles them, And then that way you can get billions and billions of different sequences, and that's what makes the receptors. Now then, once you've got your specific receptors, they can change to accommodate mutations in the pathogens, all right? So B cells and lymph nodules rapidly mutate, creating new sequences. So once you got a B cell for hepatitis is a virus. Viruses mutate constantly. Well, not only is the virus mutating, but the B cells for that hepatitis virus also mutate. And so, yeah, I mean, you're never guaranteed that they're going to mutate in the same way the virus mutated, but at least it gives you a better chance, all right? The B cells are constantly mutating, trying to keep up with the mutations in the virus itself, all right? So that's how they can adapt to newly mutating antigens. Is that clever or what? That's just amazing. Your body is so fabulously creative in coming up with ways to try to deal with the changing environment of bad guys out in the world and all the different kinds of bad guys that exist. Your body is able to accommodate that and does quite well. I mean, look, most of us live. I mean, most of us are still alive. We haven't been killed by bad guys yet. Our immune systems are really pretty darn good. Okay, so let's look at the different kinds of antibodies. As I said before, they're unique in various ways. We have 5 different types of antibodies. So remember Madge, you know the woman's name, Madge. That's the different antibodies. So we have IgM, IgA, IgD, IgG, and IgE, all right? And I know this is, there's a lot of detail here, but I want you to know that I'll try to point out what I think is kind of like the most unique or most important thing about each class, okay? This is going to be, they're being classified by the amino acid sequences of that constant region, okay? So IgM is a pentamer. So look in the upper illustration. Pentamer means has 5 parts. So look at the IgM. See, it's 5 Ys that are all basically, bolted together at the center, all right? These are the first ones to respond during your primary immune response. So when you first get invaded for the very first time by a new bad guy, by a new pathogen, IgMs are the ones that spring into action. And can you see why? Why would that make sense? IgMs have 5 arms. Each arm has 2, so each stem has 5 stems. Each stem has 2 arms. That means an IgM can grab a total of 10 bad guys. So they're the most effective ones to have because they're grabbing bad guys. They grab more per antibody than any other antibody can grab, okay? They're powerful agglutinators. They also fix complement. So the agglutinators, that means it's going to be easy for eosinophils and natural killer cells to find them and kill all the things that are attached to their arms. So IgA is a dimer. That means it has 2 parts. So the upper illustration doesn't show that, but the bottom one does. So this is like 2 Ys that are stuck together at their ends, at their stems. So these are found in a variety of bodily secretions, including saliva, sweat, tears, milk, and intestinal secretions. So what this means is you're secreting these IgAs to try to kill things before they even get into your body, right? I mean, look, saliva, sweat, tears, they're bad guys getting on you all the time. And once again, it's really part of your first line of defense. You're trying to prevent them from making it any further. And notice how they're in milk. Yeah, so in other words, when mom breastfeeds, mom is giving the infant IgAs. On Mother's Day, make sure to send your mom a card saying, Mom, thank you for the IgAs. Because remember, infants, their specific immune system isn't ready to go. It doesn't get trained, it gets trained all throughout their childhood. It's not ready to go at birth. So mom is trying to help you out. Mom is giving you her IgAs through the breast milk so that you'll be better able to fight off bad guys before your own immune system can spring into action. IgDs, these are monomers, so this is a single Y. These are the ones that are on B cells, all right? B cells have IgDs all over the outside, and then when B cells clone into plasma cells, this is what the antibodies are, all right? It makes antibodies that are identical to the IgD, all right? I mean, to the receptors. The IgD antibodies are identical to the receptors on the B cells. Then IgGs, these are monomers again, so these are just single Ys. These are most, by far, of your circulating antibodies. So once you have beaten a bad guy, all right, you continue to keep antibodies in your blood, because the fact that you got invaded by it once, means maybe there's a higher probability that you're going to get invaded by it again. So once you successfully beat it, your immune system doesn't immediately dump all the antibodies. That would be ridiculous. I mean, let's keep them around. Let's try to keep protecting ourselves in case they come back again, okay? So you've got lots of IgGs circulating through all the fluids of your body, looking for guys that you've already beaten, and then hoping to limit their effectiveness the next time they get in. These will also cross the placenta to the fetus. Look at what mom did for you once again. She gave you her own antibodies before you were even born, so that the moment she squirted you out, you already had some of these antibodies in your body fluids. And prominent in the secondary immune response for complement fixation. So again, these are the ones that when you get invaded a second time, you have the memory response. The IgGs are the ones who are mostly involved in that. And then the IgEs, again, these are monomers, single Ys. These are the troublemakers. These are the ones that are found on mast cells and basophils, all right? And they're going to stimulate the release of histamine, all right, to produce inflammation, so you can help fight the bad guys. These are the ones, though, that have a tendency to sort of overreact, all right? So the people who are allergic to bee stings, you got stung by a bee, the basophils and the mast cells start releasing massive amounts of histamine. You get massive vasodilation, PVR drops, mean arterial pressure drops. You go into anaphylactic shock. You're not able to pump blood to your tissues, and that's a life-threatening situation. So these are the ones that are good, but they can also overreact. And when they do that, that's bad, all right? So it stimulates release of histamines, attracts eosinophils. These are responsible for those immediate hypersensitivity reactions. These are when you kind of have that overreaction, and that's the troublemaker, okay? So there you are. I would like you to know basically what they do, okay? And here's a great slide from a book. Says essentially the same thing. Shows them all, talks about them. All right, so we've mentioned before that one of the great features of your specific immune system, your adaptive immune system it's also called, is that you have this memory response. That's called the anamnestic response, all right? And so here you see, look on the left side of the illustration. Notice IgMs are the first to respond. IgGs are the ones that hang around later. They're most of your circulating antibodies. So notice it says days from first exposure to antigen. So like when I was a little kid, I got the mumps. They didn't have a vaccine for that yet. Now you got the MMR, measles, mumps, rubella. Little kids don't have to get the mumps anymore. I got the mumps when I was a kid. Nasty. All right, it attacks your parotid glands, which we'll see when we get to the digestive system. Those are big salivary glands that are kind of like in front of your ear. So your cheeks swell up. You look like a chipmunk. You look like one of those, rodents that, you pack food in your cheeks. Your cheeks are all swelled out. You run a fever. You just feel like crap. And that lasts for, a week or so, all right? So notice, look at the IgMs, all right? See how, they're going up, and after a week or so, they peak. That's about when you have defeated the mumps, all right? And then now that you're better, the IgMs, they go down again, all right? And then notice that what happened was you made the IgGs later. The IgGs stick around a little longer, and then they go down as well, but you still have them, okay? So that's the initial response. But then look at the anamnestic response. Look at the secondary response. Look what happens on day 0, somewhere day 0, day 1, when you get infected by that same pathogen. So in other words, once I had the mumps and I got over the mumps, it's not like the mumps wasn't out there in the world anymore. The mumps was everywhere out in the world, all right? And the mumps virus kept getting into my body. I mean, day after day. I mean, that's just the way it is. So, over the coming weeks and years, I was still being exposed to mumps as a little kid, playing with other little kids. I was getting exposed to the mumps. But what happens is my anamnestic response. I've now got the memory cells, I've got these IgGs. So what happens on the 2nd and the 93rd and the 471st and the 76,293rd time the mumps tried to get into my body? That secondary response, that anamnestic response, kaboom, came down like a sledgehammer, and I didn't even get sick. That's really cool, and that's what we mean by immunity, all right? We have immunity. So specific immunity, methods of acquisition, how can you become immune to something, all right? How do you actually get immune? Well, there are 4 ways. Let's take a look at them right now. So active immunity is where you make your own antibodies. So in other words, you actually get infected by something, or you get exposed to something that mimics an infection. This is where you now are going to manufacture your own antibodies. These may last a lifetime. This kind of immunity might last your entire life. Since you made them, they stick around better. So we have what's called natural active immunity. See, we're going to put these together that way. So take the type here, natural, and then add that to active immunity. So natural active immunity, this is where it means you actually get sick. So like I have natural active immunity to the mumps, all right, because I got the mumps. So I made my own antibodies, and it was from natural exposure to the mumps virus itself. Natural active immunity. Now the problem is some things you don't really want to get them because they might cause severe damage, like rabies. You basically don't get natural active immunity to rabies because it kills you before you ever have a chance to beat it. Rabies is 100% fatal if you don't do something about it, all right? And I mean things like polio as well. Polio, I mean you might beat it, but it might cause lasting damage to your body. So wouldn't it be great if there was some way we could generate our own antibodies without actually having to get sick with it? Well, yeah, artificial active immunity. This is exposure via a vaccine. And if you had to list, the most important advancements in the history of medical science, vaccinations have got to be probably in the top 3, I would say. Anesthesia, antibiotics, and vaccines. I mean, holy smoke. I never had polio, but I'm immune to polio, all right? And I'm immune to polio because I have a vaccine. The vaccine caused me to make my own antibodies against polio, and therefore they are lasting potentially a lifetime, all right? Normally what they do is they take like a virus and they kill it. You can't really kill viruses because they're not alive, so you can attenuate it. Think of it basically, it's like you put it in the microwave and you cook it, so it doesn't really work anymore. And what you do is you put that attenuated virus, they call it, basically an inactive virus, into your body. What happens is it doesn't make you sick because it's been inactivated, but your immune system still recognizes it, and therefore it mounts the immune response, and therefore you get immunity. And right now, the coronavirus vaccines, the 2 main ones that are out there right now, Pfizer, which I got, and then Moderna is the other one. That's a new technology, and it's a new exciting technology. They're thinking that this is the future of vaccines because they don't even have to use an attenuated virus. You don't even get any pathogens put into your body with this vaccine. Instead, they take a tiny little bit of mRNA, messenger RNA, that they make in a lab. It doesn't actually come from the real virus. They manufacture it. And then when they put it into your body, it's not actually a pathogen, so you can't get sick from it. But what it does is it causes the cells near the site of the injection to make the little spike proteins, they call it in the coronavirus. In other words, it makes the epitopes. It causes the cells in that area to make epitopes of the coronavirus. But you don't actually get the coronavirus in you, you just make the epitopes. Then your immune system responds to the epitopes, and it launches a full-scale immune response. And by God, you get immune without ever even having the chance to get sick. Sometimes with the attenuated viruses, people do actually, it's very rare, but it can happen that people actually get sick with the disease itself. There's not even a chance of that happening with mRNA. This is the most ingenious breakthrough in the history of vaccines. They're already talking about going back and redoing some of the old vaccines as mRNA vaccines. This is phenomenal technology. And I think it's very, very likely that the people who figured this out, there's a Nobel Prize in the future waiting for those people, because this is the most important breakthrough in the history of vaccines. Now, there's also passive immunity. So what does passive immunity mean? It means you don't make your own antibodies, you get somebody else's, okay? So from another person or from an animal. Now, in this case, though, it's not going to last forever because these antibodies are not normally part of your body. You didn't make them. So what's going to happen is your body's going to see these things and it's going to get rid of them because it says, that's not us. That's not supposed to be here. So passive immunity is very short term. This is basically for emergencies when you need to get immunity right now. You haven't got time to wait for your own immune system to crank up the response. You need immunity today. And so you're willing to use the passive immunity, the artificially acquired antibodies, knowing that the immunity is not going to last. They're going to go away in no time at all. So we talk about natural passive immunity. Basically, the only way this happens is mom. Well, yeah, mom. Either while you're still a fetus, so across the placenta. Remember what kinds of antibodies were involved there? Coming across the placenta into the fetus? Those were the IgGs. And then also newborns via breast milk. Which antibodies were those? IgAs, all right? So mom, 2 times during your existence, mom gave you her own antibodies. So those were designed to help protect you, but they don't last your whole lifetime. They're only short-lived. They're eventually going to be cleared out of your body. All right, so that's natural passive immunity. And then artificial passive immunity is where you get the injection of an immune serum. Classic examples of this would be for rabies or for snake bite. So some people, now you'll be able to spot this. Some people mistakenly say that, like, if you're bitten by an animal and they suspect rabies, you have to get a rabies vaccine. No. Rabies vaccine would be worthless in that case. It won't. A rabies vaccine would be a form of what? Think about it. What would that be? That would be artificial active immunity. But your immune system won't have time to kill rabies before it kills you. So what you need is rabies anti-serum, they often call it, or rabies immunoglobulin. It's not a vaccine. You're actually getting the antibodies themselves. You are having antibodies put into your body. You're not making them. You're getting them from outside, right? And then the same thing with a rattlesnake bite. What you're going to do in this case is you're going to get antibodies that have been manufactured, usually by either a horse or a sheep. In the case of rabies, they actually use humans, all right? So there is a rabies vaccine. I have friends who work in animal rehab. One of them she's called the bat lady. She works with bats. Bats are the #1 vector for rabies. They're the main carriers of rabies. Even though hardly any bats have them, they're still the #1 source of rabies. So she gets a rabies vaccine, okay? So the rabies vaccine causes her to make her own rabies antibodies. Therefore, she can comfortably work with animals that might be rabid because she's immune. But what they do is then she actually donates her blood plasma and they make the rabies anti-serum from people like her. She got the rabies vaccine, she makes the immunoglobulins, and they take immunoglobulins from her. Snake, they do either sheep or horses. What they do is they have like big stables full of sheep or horses. They inject them with tiny amounts of snake venom, not enough to make them sick really, but enough so that they make antibodies. And then they take blood from those animals and they centrifuge it down and go through a bunch of steps and they extract the antibodies from their blood plasma, okay? Make sure you get those straight. I'm going to ask you about these. Natural active immunity, artificial active immunity, natural passive immunity, artificial passive immunity, all right? There is one other way to get immunity, by the way, and that's essential oils. No, for God's sakes. Okay, so this is just a little bit of a sidetrack here, but you know, it's relevant. This is a beautiful place up in southern Utah called the Paria Valley, southern Utah. Um, there was a town called Paria. It's where they shot the film Outlaw Josie Wales, the last half of the film. The town has since been destroyed. It was vandalized. It was intentionally set on fire. Hard to believe. And then they actually rebuilt it, and then they vandalized it again. So I don't know. But at any rate, this is near that town of Paria. And near the town, you can see there were settlements there back, in the cowboy days. And there's a huge graveyard. It's a communal graveyard there where they've got lots of the people who lived in Perea back in the late 1800s. That's where they're buried. But I took a look at some of the plaques here. So look, in memory of, and you see all the names. You see born and died. So like the first one, 1815, 1892. Okay, great. That person lived a long life. Lived to be 77. 1813, 1897. The person lived to be 84. 1866, 1880. That person only 14. 1882 to 1882. Yeah. That person didn't live long at all, did they? And if you look down the list, 1877 to 1879, 1891 to 1891, 1878 to 1879. There are a lot of people there who didn't live very long. I don't know what killed them, but one of the things that used to kill little kids like crazy were diseases, all right? Things, rheumatic fever and stuff like that. All kinds of diseases that we didn't have antibiotics for, and we didn't have vaccines for either. There was a time in the history of humans where childhood diseases killed tons of little kids, all right? That doesn't have to happen anymore because of vaccines. Vaccines let me consult my coffee group friends because medical professionals know nothing. The diseases aren't deadly, and it's better to have natural immunity instead, said no parent in a developing country ever. The number of lives that have been saved around the world by vaccines is phenomenal. Vaccines, again, one of the most significant advancements in the history of medical science. Measles, the decline, 99% decline in measles, 93% decline in pertussis. Polio, for all practical purposes, wiped out. Rubella, 99%. Diphtheria, 100%. Mumps, 99%. Just showing here pre-vaccine morbidity, recent reports. Look at this. And seriously, on the right-hand side, you see how many people are dying now that we have vaccines? Basically none. How many people died before vaccines? Tons. Tons of people died before vaccines. And really, on the right-hand side, a lot of those things would be down to zero, except you've got these nutcases out there who don't want to get vaccinated. There you go. There's your flu shots right there.